Hi everyone who is currently watching my poster presentation for the second edition of the Women in Bioinformatics and Data Science LA conference. And in this presentation, I will tell you a little about my scientific initiation project, which I carry out at the Brazilian Laboratory of Science Technologies, called Development of a Pipeline of Genomic and Transcriptomic Data Analysis for the Identification of Target Genes for Silence, RNAi, of the pathogen M. incognita. Well, in my research, as evidenced by the title, we use the gene silence technique employed by RNAi interference technology. With this in mind, nowadays this mechanism has been used a lot, in which is conserved by evolution in other life forms to control agricultural species through the knockdown of essential genes in the parasite. This species has a very cosmopolitan behavior, having greater reach in the tropical region of the globe, generating a loss of $157 billion annually. Every year in Brazil, there is a loss in the order of 35 billion reais to fine nematodes such as the incognita. This nematode has four life stages: egg, juvenile, J2, J3, J4, and adult stages. The infective process became irreversible in the J2 life stage, such that at the time the juvenile peach is gradually feeding on cells from the root of the cultivar in order to develop, then finally producing the characteristic symptoms of infection by this species, the production of galls in the roots. In this context, differently expressed genes in J2 are our target for silencing. Furthermore, these genes must be in a single copy in a genome. It must be conserved within the genome of different studies it isolates in order to guarantee a wide applicability. Here in Figure 2, you can check an image showing the damage done to the roots of a cultivar during maturation in the J2 life states of M. incognita. To carry out this careful selection of genes, we propose to develop a pipeline to optimize the search for target genes for silence by RNAi. To do this, we use public genome and transcriptome data of the target nematode. So here in Figure 3, we can follow the diagram that contains the step-by-step of our pipeline, as well as each software used. This figure shows a process that range from the assembly and quantification of transcripts, followed by the analysis of difference expression and clustering by expression cluster, also contemplating our priority object of our searching for single copy orthologous genes, conserving the various sequence isolates. Among the main results, we initially evaluated the transcriptome assembly using reference genome in comparison with the de novo strategy. In order to decide which genome to use, we compare some metrics, such as the number of coding transcripts detected and the value of N50, which can show us the amount of proteins produced during the nematode's life stage and the quality of assembly of each genome, respectively. We conclude that the reference genome would be the best choice for our further analysis, since this one represents the higher value of the genome, again for both parameters, as you can see in Figure 4. And lastly, we perform the cluster of all genes along the genome that are differently expressed for each nematode life stage. So as you can see in Figure 5, I have highlighted the G2 target lifestyle for all generate sets. We can now pass these genes through the filter of the established criteria for target gene selection and select only a few gene sets, such as sets 4 and sets 10, which present a significant expression in the target life stage, J2. Our next steps are focused on grouping these genes previously selected by the cluster into orthologous and paralogous, discarding the paralogous since we only want genes that have a single copy in the organism. After that, we will verify the presence of SNPs, variable regions in the genome, and election and insertion mutations in order to select only the most conserved genes among the samples obtained from Brazilian cultivars. From this, we think that it is possible to group together a few well-selected genes that meet all the criteria, and that will later be silenced in the M-incognita gene at the laboratory stage via RNAi. Finally, we will analyze the effects of the absence of these genes in relation to the infective process of this nematode, in which we hope to have innovative results to add to the methodology for prospecting target genes for agriculture pest control, as well as to the success of this project in gradually terminate the appearance of M. incognita in cultivars around the world. Thank you very much.